Hope you had a nice weekend? No? That was a yes, I guess, right? All right, so we are moving along. It's like somebody forgot their eraser. And today I want to finish up talking about nucleotide metabolism, and then we'll start talking about replication of DNA. For those of you looking for regrades, I haven't done the regrades yet. They'll probably be available in the BB office midweek, I would say. So, I've also been asked about doing an overview session, which had completely slipped my mind. You guys know how that is when you get to be my age. Maybe you don't, but I can tell you how it happens. In any event, I will do one. I'll look at the calendar. I'm thinking maybe Wednesday night, but I've got to look to see if I have anything scheduled. And I will videotape it as always. Okay. Well, last time when I was talking, I finished with this spot right here. And this shows the overall reaction catalyzed by ribonucleotide reductase. As I will show you in a couple of minutes, the mechanism for this is fairly complicated. I'm not going to hold you responsible for much of the mechanism, just little pieces of it. But suffice it to say that what the enzyme is catalyzing is essentially the removal of an oxygen off of a ribonucleoside diphosphate. That's an important thing. Ribonucleotide reductase works on ribonucleoside diphosphates such as ADP, GDP, CDP, and UDP. And the product of that then is dGDP, dCDP, dADP, or dUDP. Now notice thymidine has not entered the picture yet, and we'll see later how thymidine enters the picture as a result of additional metabolism of uridine nucleotides. So, dUDP is in fact made. And you'll also see that this reaction, which, as I said, is a bit complicated, removal of an oxygen, is in essence a reduction. And whenever we have reduction, we have to have a source of electrons and protons. And the source of electrons and protons for the reduction of ribonucleotides ultimately comes from NADPH through thioredoxin. So thioredoxin donates electrons to make this happen. It gets the electrons replenished by electrons and protons from NADPH. So as a result of that, we're able to synthesize nucleoside diphosphates. And I hope you'll remember from what I told you last time how dCDP, for example, would be made into dCTP. What enzyme would do that? It's Monday. Yes? What enzyme catalyzes the conversion of dCDP into dCTP? The whole class doesn't know that? Wow. Yes? NDPK. NDPK works on every diphosphate. Every diphosphate. Extra point of credit right there. Right? dCDP is a diphosphate, right? And NDPK works on all the diphosphates, converts all of them to triphosphates. Consequently, dCDP gets converted into dCTP. Similarly, dADP gets converted into dATP, dGDP into dGTP, and dUDP into dUTP. Now, while I'm on the subject of conversion to phosphates, I want to say a little bit about dUTP. dUTP is a problem for a cell. Why is a cell even making dUTP? Well, the answer to that is that the cell needs to make dUTP in order to make thymidine nucleotides. It's also hard to restrict dUDP from the NDPK because NDPK has a very broad specificity and it's able to take in dUDP and make dUTP. Well, the problem that dUTP poses for the cell is that DNA polymerase will recognize dUTP as a nucleotide just like it recognizes dTTP, thymidine. Okay? So consequently, cells can get trace amounts of U's into their DNA. That's problematic, right? Fortunately, there are two mechanisms, as we will see, that help cells to avoid doing that. One mechanism that we'll talk about here relative to nucleotide metabolism actually relates to an enzyme called dUTPase. dUTPase. Okay? dUTPase is found in all cells, and dUTPase has the function of converting dUTP into dUMP, dump. When it's in the dump form, dUMP can't be made into DNA and no longer poses a problem. Okay? So dUTPase is very important, one of two things, for keeping U out of DNA. The other one we'll talk about later when I talk about repair mechanisms that cells have to deal with U. Okay, questions about that? Okay. Now, this is our enzyme, ribonucleotide reductase. As I said, you can call it RNR. And ribonucleotide reductase has two subunits called R1 and R2. The R1 subunit is referred to as the large subunit of ribonucleotide reductase, and R2 is referred to as the small subunit of ribonucleotide reductase. The catalytic action of ribonucleotide reductase is contained on the large subunit. Similarly, the allosteric regulation of ribonucleotide reductase is also carried on the large subunit. So the large subunit has several important functions. What's the small subunit there for? Well, the small subunit is there to provide a way for this, the enzyme to generate a radical that's needed for the catalytic mechanism of the enzyme. Now, this is an unusual and interesting process, and it happens in most, not all, but most of the ribonucleotide reductases that have been studied. Okay? There's a tyrosine side chain in the small subunit of ribonucleotide reductase that loses a proton, okay? Loses a hydrogen, actually. Loses a hydrogen, and as a consequence, is radicalized. That radical is essentially electronically communicated all the way through the small subunit and into the active site of the large subunit. When I talk about these proteins being circuits, you can really see how that happens with ribonucleotide reductase, because not only is that radical communicated all the way through a subunit, but it actually jumps subunits into the large subunit so that it can catalyze the reaction that I showed you on the last slide. Okay? So, that radical on the tyrosine is very, very critical. It's essential for most ribonucleotide reductase enzymes that's out there. Okay. This schematically depicts the ribonucleotide reductase. You can see it's a dimer of dimers. The R1 subunit is the large subunit. It has the active site. It also has the specificity site. And it has something else called the activity site. So it's actually got 3 sites on it. And it's a very unusual enzyme in this respect. Okay? The R2 is where the radical is generated. And the radical is generated and then essentially communicates with the active site to allow the catalysis to occur. Now, the mechanism that this enzyme goes through, as I said, I'm not going to hold you responsible for the details of the mechanism because it's a fairly complicated mechanism. But we can see the tyrosyl radical that is ultimately up here. And this tyrosyl radical plays this role in catalysis. The tyrosyl radical is involved in the movement of electrons and in the movement of protons in this overall process. During the process, this hydroxyl group right here gains a proton. And when it gains a proton, it's got H2O with a plus down there on it. And it's the loss of that water that actually causes this molecule to lose, that's how it loses its oxygen. And you'll see what's left behind is the plus sign that was on the H2O. So the water comes off, taking the electron with it, leaving the plus sign behind here. And that plus sign must gain a hydrogen in order to complete its, to get to the finished base right here, the finished nucleotide right here. In this process, this tyrosyl radical is donating the hydrogen that's going across here and creating this reduced ribonucleotide that you see here. Okay. What else do I want to say? I think that's it. So it's a complicated reaction. Now, what's more interesting from my perspective at least is the regulation of ribonucleotide reductase. Ribonucleotide reductase has a very complicated and unusual regulation system. And based on what I've been telling you all about the balance of nucleotides, it shouldn't come as any surprise that the cell has some very elaborate controls to make sure it's not making too much or too little of any given deoxyribonucleotide. Because, of course, it's the deoxyribonucleotides that are used to make DNA. Another consideration here is, of course, DNA is the blueprint. And we take for granted that fact. The DNA is the blueprint. We don't mess with the blueprint. We can mess with what's made from the blueprint. We don't care, and we'll see this when we talk about transcription, we don't care nearly as much about mistakes that occur in making of RNA as we do of mistakes that occur in the making of DNA. Because RNA isn't around forever. If you make a mistake making an RNA, it might make a non-functional protein for a while, but that RNA won't be sitting around forever. And cells also make more than one RNA of things anyway. So if they make a mistake in one, then another one makes up for it. With DNA, it's essential, however, that the cell make as few mistakes as possible. So, the regulation of ribonucleotide reductase then is very, very critical to understanding what's happening with balancing of all those deoxyribonucleotides. Well, let's start with the simple part of this enzyme. Now remember, we're only looking at the large subunit. The large subunit has the active site, it has what I call an activity site, and it has a substrate binding site. It has an allosteric site. Okay, well, if we start with the activity site, let's first of all define these terms. What's an activity site compared to an active site? The active site, as is true for any enzyme, is the place where the reaction is catalyzed. So the active site is where the reaction is catalyzed, and it's the active site that binds diphosphates, because you remember that diphosphates are used to make the deoxyribonucleotides. So the active site has binding site that will accept any of those 4 nucleoside diphosphates. And the activity site is an allosteric site. This enzyme actually has 2 different allosteric sites. The activity site is the easier to understand of the 2. If ATP binds to the activity site, the enzyme is activated and deoxyribonucleotides will be made. That is, the active site will accept diphosphates and create deoxyribonucleotides. This sort of makes sense, because remember how we've talked about the cell has to make decisions in terms of, am I going to divide or not? And some of those decisions include, do I have enough energy to divide, right? So it's not a surprise that the cell would have a switch that says, hey, we've got enough energy, it's okay to go forward. On the other hand, dATP, oh, question? Yeah, Leo.
It looks like they're flipped up there, because it's trying to activate the dATP.
Oh, it does say that. Yeah, good. Okay, yes, you're right. One of the good things that's happened with this class is we found a lot of different typos that are not typos, but actually mistakes that are in the figures. That's a mistake on the figure. That should be, that should say dATP, that should say ATP. Yes. Thank you. You know, the thing is, as I've said before, you stare at a figure. I've been looking at those figures for months, if not years, and until you stand in front of a bunch of people with it, you don't see it. I didn't see that, but that's exactly right. Okay? Now, ATP activates the enzyme. dATP inactivates the enzyme. Why does dATP inactivate the enzyme? Because dATP indicates that the cell has plenty of dATP. There's plenty of dATP. Now, dATP is the signal that says all of the nucleotides we have plenty of. Why do we rely, this is a common question I get, why do we rely only on one nucleotide to tell us that? Okay? Well, if we balanced all of the nucleotides, then reading any one of them will tell us if we have the right amount. Okay? So dATP turns this guy off, ATP turns this guy on. Okay. Yeah, another question, yeah.
What's the difference between an activity site and a replication?
Okay, so I call this an activity site because it determines whether or not the enzyme is active. It's an on-off switch for activity. It is an allosteric site, to be sure. But I call the regulatory or specificity site by a different name because, as we'll see, it doesn't work in the same way. Okay? Okay. The substrate specificity site, also called the regulatory site or allosteric site, is very interesting in this enzyme. Okay? Now, I'm going to do some simplification for it, but I'm also going to show you all of what happens here. So if we look at this, we see there's 4 different molecules that can bind into this substrate specificity site. They're all triphosphates, just as these were both triphosphates. The only place we have diphosphates is at the active site. At the substrate specificity site, for example, if dTTP binds, okay, it favors the binding on the active site of GDP. Why? Well, let's think about this. dTTP is a pyrimidine, and excess pyrimidines mean that you would want to make purines because they're the complementary bases to pyrimidines. So binding of a pyrimidine favors the synthesis of purine deoxyribonucleotides. This is a balance thing. Okay? If ATP or dATP bind, that favors the synthesis of pyrimidines. So binding of this purine, these purines will favor the synthesis of pyrimidines. And finally, dGDP, if it binds up here, will favor the binding of ADP. Now that one kind of breaks the rules. Okay? But suffice it to say, despite the complexity that's there in terms of the allosteric effectors, despite the complexity that's there, the cell balances the relative amounts of deoxyribonucleotides it makes beautifully. Okay? It balances those beautifully so that mutation is not a problem. If you don't balance the nucleotides properly, you can increase mutation rates by 100 or 1,000-fold. Okay? Making mutations in your blueprints will not work. Now, the simple rule to understand this is binding of pyrimidines up here favors purines down here. Binding of purines up here favors pyrimidines down here. All right? So I'm not going to ask you, if you bind dTTP, which specific nucleotide is favored down here. I think that's just an exercise, and that's a ridiculous exercise. But understanding the principle of pyrimidines favors purines and purines favors pyrimidines is what I think that you should know. Was there a question? Yes? So up here, the binding of dGDP favored the synthesis of dATP, or dADP, right? So there's a purine favoring a purine. That was the exception. Okay? Other questions? Okay. Well, so So with ribonucleotide reductase, we can make all of the deoxyribonucleoside diphosphates, and with NDPK, we can convert all those deoxyribonucleoside diphosphates into deoxyribonucleoside triphosphates. So now we can make DNA, except for we haven't made thymidine. Okay? So thymidine we have to make from uridine nucleotides. The scheme by which the cells go about this is interesting and odd, although it sort of makes sense given the enzymes that cells have. Okay? If we start with UDP, UDP is a precursor of dUDP, catalyzed by ribonucleotide reductase. NDPK, with its broad specificity, will convert dUDP into dUTP. And then, as I told you, dUTP is converted into dUMP by dUTPase. That's essential to keep the U's out of the DNA as much as possible. Now, there are 1, 2, 3 more steps to get to dUTP. I'll show you these in more detail in a second, but suffice it to say the first one involves converting dUMP into dTMP in a reaction catalyzed by the enzyme thymidylate synthase. That reaction involves addition of a methyl group. This donor over here, a folate, is a common donor of single carbons to molecules. Okay? So the methyl group that comes from here comes from this folate, and that's what makes dTMP. Okay? The enzyme thymidylate synthase, clearly, critically an important enzyme. It's a target in some cases for anti-cancer drugs, and I'll show you another example of that in just a second. To go from dTMP to dTDP, that is to go from the monophosphate to the diphosphate, we've got a specific kinase, dTMP kinase, that catalyzes that reaction. And then, of course, to go from dTDP to dTTP, we use the enzyme NDPK. So as a result of this long, convoluted pathway, thymidine nucleotides can be made that can then be incorporated into DNA. Okay, so here's the reaction that I described to you. You can see the addition of the methyl group going on right here, the folate donor, and the critical thing about this reaction is actually right here. What is left of the folate after it's donated this methyl group? Well, it turns out that the folate is converted to a form called dihydrofolate. It's different than the original one since obviously it gave up a carbon. Okay? That dihydrofolate has to be converted back into a tetrahydrofolate. This is what the THF stands for here. It has to be converted into a tetrahydrofolate if the cell is to continue to make thymidine nucleotides. Okay? Cells have limiting supplies of any of these reagents, and so just like it only has a limited amount of NAD and NADH that have to be interconverted, so too do folates have to be interconverted. So the dihydrofolate has to be converted back into a tetrahydrofolate in order for thymidine nucleotide synthesis to continue. Okay? Here's the recycling reaction that occurs. Okay? So the dihydrofolate right here, okay, can be made into a tetrahydrofolate. And by the way, there are many folates. And I'm not going to expect you to know all of them. All I care about is that you know that the dihydrofolates get converted into the tetrahydrofolates. Dihydrofolate gets converted into tetrahydrofolate as a result of catalysis by this enzyme right here, which is known as dihydrofolate reductase. And as its name would suggest, this reaction is a reduction. It requires electrons from NADPH. Okay? And this enzyme can be inhibited by the molecule methotrexate. Now, I've talked about this molecule earlier in the term. Methotrexate is a competitive inhibitor of dihydrofolate reductase. Now, this is critical because methotrexate is used in some chemotherapies, because if one inhibits the synthesis of thymidine nucleotides, one can kill a cell. Well, since you don't want to kill all the cells, typically what you do with this is you give a patient methotrexate for a short period of time, and during that short period of time, the cancer cells that are dividing more rapidly are more susceptible to the treatment of this drug than are the normal cells. Then you withdraw the drug, allow the other cells to recover, and hopefully you've killed a lot more cancer cells than you have normal cells. Okay? So this enzyme is critical in that chemotherapeutic process. Now the other reaction that's on here, I just simply put it on here to show you how amino acid metabolism, which is what this is right here, is involved in interconverting folates. And that's just, there's nothing specific about that I'm going to expect you to know. Okay? Now, I do want you to understand that this enzyme right here, okay, it can be targeted in treating malaria. So it turns out that the organism that causes malaria has a slightly different form of this enzyme that's different enough from the human form of the enzyme that it can be targeted specifically to kill the plasmodium that carries that and not affect human cells. And so that's one kind of a cool aspect of folate metabolism. Okay, questions about that? All right. Now, another enzyme that I talked about that was important in terms of chemotherapy is actually thymidylate synthase, and that's going directly to the source of the thymidine nucleotides. Thymidylate synthase is inhibited by this compound called 5-fluorouracil. And 5-fluorouracil looks enough like this part of the ring that's on dUDP that it binds to the enzyme covalently and acts as a suicide inhibitor, preventing the enzyme from functioning. So by doing this, you can imagine this might be a fairly radical treatment, because if you did this to all cells, you would kill the patient, and that's not considered good in the old medical trade. Consequently, it would obviously be targeted to a specific set of cells in its use. Okay. And one more time to remind you, I think I've shown this a couple times in the course, but there's methotrexate, competitive inhibitor for dihydrofolate. They look very, very similar, and not too surprising that that methotrexate can inhibit the enzyme. Methotrexate is a competitive inhibitor. It's not a suicide inhibitor. Yes?
Is DHFR the abbreviation that we could use?
Yes, I'm sorry. DHFR is an abbreviation for dihydrofolate reductase, and you're welcome to use that abbreviation. You're also welcome to use DHF for dihydrofolate. So, yeah. Don't confuse the two. One's an enzyme, one's not. Okay. And a whole bunch of points there with respect to nucleotide metabolism. Anything on there that I haven't gone through specifically? As I said, understand the pyrimidine, purine business here, and you should be in good shape. Okay. Good summary there. All right, the last thing I want to talk about actually has to do with, again, some therapeutic medications that are out there. And these are nucleotide, and in some cases nucleoside, and in fact, almost in every case, they're nucleoside analogs, and there's a reason for that. So let's first of all look at their structures, and then I'll talk about why we use nucleoside analogs instead of nucleotide analogs. So there's AMP and there's adenosine, and the difference between AMP and adenosine, of course, is that phosphate right there. Phosphate, if I want to give you a drug that I want to get into your cells and kill that cell, for example, then the biggest limitation I actually have is getting that drug into your cells. And it turns out that when you attach a phosphate to something, it doesn't get into cells very well. There's no transport systems that allow those things in very readily. On the other hand, if the phosphate is off of there, then what you see is that this guy here will make it much more easily into cells. Well, of course, adenosine is not a drug that I would use to kill a cell, because the cell would take it in and make AMP out of it, and then ADP, ATP, so it wouldn't be any problem. But that's actually the strategy for each of these drugs. Each of the drugs that I will show you here is, in fact, a nucleoside analog that the cell takes in, phosphorylates, and then it gets stuck. Okay? So the trick is to give it to cells as a nucleoside, let it phosphorylate it, so the phosphate will go on here, just like you can see. And then, when the enzyme, let's say an RNA polymerase that would otherwise use it, can no longer grab it and do its thing. So that will kill the cell. Okay? Alright, so look at these. If you look at these, you see a variety of analogs, some of which are like we've already talked about. Here's something that's got hypoxanthine up there instead of guanine. Well, you remember that hypoxanthine looks a lot like guanine, but you'll see that it also lacks a 3' hydroxyl. We'll talk later today, actually, on the synthesis of DNA, how a 3' hydroxyl is necessary for growing a DNA chain. So if you take this guy and you have it on the triphosphate form, and the polymerase grabs it and it attaches it to the end of a DNA strand, nothing additional will be able to be put onto it because there's nothing to attach the next one to. So things like this are called chain terminators. They will lack the ability to connect the next nucleotide at the 3' position. In this case, because it lacks an oxygen. It lacks a hydroxide. Okay? Okay, so DDI, you probably have heard about. DDI is used to treat HIV. It's one of the cocktail drugs commonly used. And it specifically will inhibit the synthesis of DNA. It turns out that the HIV polymerase is more susceptible to this drug than our polymerase is, and consequently, HIV gets stopped as a result of that. Whenever you're designing an antibiotic, you're always looking for that antibiotic to specifically kill the cells that you don't want, while not affecting the cells that you do want. That's why chemotherapy is very difficult to treat, because again, in chemotherapy, cancer cells are just like our normal cells with a slight tweak, but they have the same enzymes and so forth in them. Okay? So, DDI is useful for that purpose. What else? What else do I want to say here? Vidirabine, Era A, okay, similar strategy here, except for the fact that we've got a ribose there in the sugar instead of ribose. Notice that ribose would have both of those hydroxyls down. Ribose has one hydroxyl up, one hydroxyl down. That messes with RNA polymerase. And because it messes with RNA polymerase, it's able to stop transcription from occurring. It also can, if it's phosphorylated, inhibit viral DNA polymerases. This drug is commonly used to treat herpes, because the herpes simplex DNA polymerase is much more susceptible to it than our own DNA polymerase is. Okay? Let's see. Here's an analog of adenosine that you can see here, and that analog has got a slight change in the bonding up here. It makes a very, very slightly different compound called BCX4430, which I don't expect you to know, by the way. And it's a competitor of ATP, wherever ATP is used. So you put phosphates on here, you can think of all the different places in cells where ATP might be used, but if it's mucking up the enzyme's ability to transfer that phosphate onto another molecule, you could have very drastic effects inside of cells. As you can see, it's used to treat Ebola. And last but not least, there's a bunch more. And I'm just showing you these. I'm not asking you to memorize the names of these or how they work. Just an idea about how nucleoside analogs can be used to treat various compounds. Some of these are pretty exotic. Look at this guy over here, lamivudine, 3TC. 3TC is also used as an anti-HIV. And you can see that it's also a chain terminator. It has a sulfur down in that position right down there. There's a chain terminator right here, DDC. This guy, I don't even know how it has any effect whatsoever. You'd think that enzymes, any self-respecting enzyme working with nucleotides wouldn't bind this guy, but it does, and it's used to treat HIV and also other viruses. Okay, and yes, and these are all deoxycytidine analogs for what that's worth. All right. Well, we have finished nucleotide metabolism. We have, in fact, finished metabolism, except for some stuff I'm going to talk about at the very end of the course. Okay? You've had enough metabolism. It's time for us to turn our attention to a new topic. The new topic relates to the replication of nucleic acids. Specifically, in this set of lectures, I'll be talking about the replication of DNA. And replication of DNA is something that you've probably already heard a lot about in biology classes, even heard about it in high school, and so forth. I start talking about DNA and DNA replication from a perspective of structure, and I'll give you some details about structure that you probably haven't gotten in some of your other courses. You know, of course, that nucleotides have a structure like here. We've already talked about this. Deoxyribonucleotides have a hydrogen down there instead of a hydroxyl, whereas ribonucleotides, okay, I just saw that one. You know, you really appreciate what copy editors do when they publish a book, because between Indira's and my eyes, we've seen these things probably 5,000 times, and still you don't notice it until there. But a ribonucleotide has a hydroxyl at position number 2. Now, this triphosphate, you know, is important for energy. ATP has high energy. You learn that. One of the things that I like to point out is that when you make DNA, it's these 2 phosphates that get clipped off. Only 1 phosphate of the triphosphate makes it into nucleic acids. And when we think about that, it might seem, okay, well, so what? Well, there's a very big so what that's almost never discussed about that. This guy right here, these 2 phosphates on the end, get clipped off during the synthesis of DNA and also the synthesis of RNA. Okay? That's called pyrophosphate. It's abbreviated PPI. There's a covalent bond between those 2 phosphates. And you might say, okay, well, you can get some energy if you break that bond. And yeah, you could get a little bit of energy if you break that bond, but that's not the reason that that molecule is important. That molecule is important because if I start, I'm going to describe the reaction to you. If I say I have RNA plus ATP, okay, goes to RNA plus an additional AM, a monophosphate on there, plus PPI, that's the product of the reaction. Okay? Everybody follow me? So I've got an RNA with an ATP. Polymerase now links the A to the RNA, so I get an RNA plus an A, and I get a PPI. That's what's happening in transcription. It's also what's happening in DNA replication. Why am I making a big deal about this? Because that PPI is a target for an enzyme known as a pyrophosphorylase. Pyrophosphorylase. Pyrophosphorylase is found everywhere in the cell. There's a lot of it. It converts PPI into 2 phosphates, breaks the bond between the 2. And by doing so, it actually is favoring the reduction in the concentration of the product, PPI. The other product, of course, was an RNA that had grown in chain length by 1 nucleotide. So what this reaction has done is it has favored the production of product. It has made that reaction be pulled. Remember pulling and pushing? Pulling occurs when you remove product. You've removed product here. This reaction is actually favoring the synthesis of RNA and favoring the synthesis of DNA. Okay? Without that pyrophosphorylase reaction occurring, this process of transcription, the making of RNA, and DNA replication would not occur nearly as readily as it does. And that's a reaction that you almost never hear people talk about, but it's absolutely essential for cells to make the nucleic acids that they make. Okay, there are the deoxyribonucleotides, as you can see, dATP, dGDP being the purines above, dCDP and dTTP being the pyrimidines below. Okay? In making a DNA strand, of course, a phosphate from one nucleotide is attached to the hydroxyl at position 3 of another. The reaction always goes in the direction of what we call 5' to 3'. And we can see that here. Okay? This examines a single strand of DNA, and it's showing that there are actually two ends to that molecule. DNA has what we call a polarity. At the 5' end, we see the phosphate up here, and at the 3' end, we see a free hydroxyl. If we were to add another nucleotide to this structure, it would go on right here. On the 3' end, a new one would be attached. So that the thing that will grow during DNA synthesis is, in fact, the 3' end of the molecule. Okay? The bonds that hold together the individual nucleotides are known as phosphodiester bonds. Okay? Phosphodiester bonds. DNA polymerase makes phosphodiester bonds. RNA polymerase makes phosphodiester bonds. Those are the backbones of DNA. The backbone of DNA we describe as being phosphate, sugar, phosphate, sugar, phosphate. And I like to think of DNA as having an inside and an outside. It's maybe simplifying that way, but that's the way I like to think about it. On the inside we have the bases. That's where the base pairing occurs. The bases are relatively nonpolar. They're relatively nonpolar. They don't like water as much as the sugar and the phosphate. Both of which are very, very hydrophilic. Right? So if we think about what happens with the double helix. What happens with the double helix is that coiling to make that double helical structure is actually doing something very similar to what proteins were doing when they were folding. Proteins folded so that the nonpolars were largely on the inside and the polar molecules were largely on the outside. Okay? It is said that Linus Pauling, the biggest mistake Linus Pauling ever made in his life, was he proposed a structure for DNA. When they were in the race to determine the structure of DNA, he proposed a structure of DNA. Because at that time they didn't know what the structure was. And he figured that the most important things were the bases. They had already figured that out. The bases were really critical. They didn't know how they did what they did, but they knew the bases were critical. And so in his logical mind, he said that the most important things will be the most readily accessible things. The bases will be on the outside. And the things that don't matter, the riboses, the sugars, and the phosphates, those are going to be on the inside. Well, from a logical perspective, that made very good sense. From a chemical perspective, it didn't make any sense at all. Well, chemistry won. Okay? Because we know today, of course, that the sugars and the phosphates are on the outside where they can interact with water, and the bases are on the inside where they can interact with each other. Okay. All right. This shows the base pairing that occurs inside of the double helix. It doesn't show the helical nature of that, and I'm sure you've seen the models of that, so I don't need to go through and describe the models. But you can see the hydrogen bonds that form between each of the bases within the double helix. Thymine, of course, forms base pairs with adenine, and the base pairs are stabilized by hydrogen bonds. So there are two hydrogen bonds between a T and an A. And there are three hydrogen bonds between a C and a G. That means that CG base pairs are harder to split apart than AT base pairs are. And that will turn out to be critical when we talk about transcription. Because when we talk about transcription, we'll see that there are sequences called promoters that RNA polymerase needs to separate the strands and get inside of. And promoters frequently have AT-rich sequences, meaning it's easier for the polymerase to get in there and separate the strands and do its thing. Okay? Another nice thing about the structure of DNA that is so beautiful is the way that the base pairing occurs, given the sizes of these molecules. We see a purine paired with a pyrimidine, we see a purine with a pyrimidine, we see a pyrimidine with a purine, we see a pyrimidine with a purine, etc. The beauty is it doesn't matter if you have an AT or a GC, the width of this double helix is almost exactly the same. So it doesn't bulge at places where you have an AT compared to a GC base pair. And that turns out to be really, really useful when we talk about recognizing DNA that's been damaged. Because DNA that's been damaged will have bulges. DNA that has mispaired bases will have bulges. And those bulges can be read, as it were, by enzymes that recognize bulges within the double helix. Okay, that's a lot of material for today, so I think I will turn our attention to the song, which is one of my favorite songs. It's a beautiful song, and I hope you will sing. I'm not going to sing it. I'm going to have this lady sing it because she's much better than I am.